Today, we are going to present a computer vision project of enhancing nucleus segmentation and its 3D reconstruction using super-resolution by Rita Suriman, Zeynep Yıldız, and Jaroslavik Pimagambetov.
We will be talking about introduction, the data, super-resolution model, segmentation pipeline, 3D reconstruction, and evaluation results. Curious to see 3D figures of cells? Be ready to be just enchanted as we were by the final results of this project. So, what is the big idea? In this project, we improved the quality of low-resolution histology images using super-resolution models. We then used those enhanced images to get better results in nucleus segmentation and 3D cell reconstruction. Why is this important? It enables quantitative 3D analysis of nuclei shape and volume, reveals special patterns that aren't obvious in 2D, helps researchers study cell organization in tissues.
Now, for the dataset preparation and pre-processing. For our dataset, we used Nucleus Seg, a fully annotated dataset for nucleus instance segmentation in H&E stained images, where we have high-resolution histology images showing cell structures along with binary masks that label individual nuclei as our ground truth. Now, although we had 14 different data type folders, our main data consists of nucleus images and are the base for segmentation and reconstruction, where the binary masks that label individual nuclei in the images, which were used as ground truth for training and evaluating segmentation models. So our pre-processing pipeline was pretty straightforward. We organized folders for train validation and test splits, selected valid samples with matching images and masks, and we split the data into 81% training, 9% validation, and 10% test. Finally, we resized everything to 256 by 256 to create downsampled versions. So we took high-quality images, we made them worse on purpose, and it was all for science. So this downsampling actually lets us test how well our super-resolution models can recover the original quality.
To evaluate the performance of different super-resolution models, we first selected a subset of the dataset and applied 6 SR models. 4 of these models are custom-trained models, and 2 of them are OpenCV-based models. Based on their performance on the subset, we selected 3 models, SRCNN, ESPCN, and EDSR custom trains. As you can see in the result, we choose the EDSR because it achieved the highest score.
Starting with our segmentation pipeline, we first extracted 256 to 256 patches from the super-resolved images, normalized them, and binarized the ground truth masks. We then trained the standard unit 4 down and the 3 up sampling stages with Adam optimizer for 30 epochs. After training, we found that 0.5 threshold maximizes both dice score and the intersection of union scores, and applied a simple connected component cleanup to remove any masks under 100 pixels. As you can see in the training graph, both training and validation loss steadily decreases, with validation dice climbing up to 83% by epoch 30 and no significant overfitting. Finally, in the segmentation cell images, the unit accurately recovers nuclear boundaries, the post-processing step cleans up stray pixels, and the yielding smoother masks with an extra gain in dice score.
Here's where things get really interesting. We're taking flat 2D histology images, like this one from a human esophagus tissue, and we're reconstructing them into complete 3D models. Now, for this one, the technical pipeline was actually a bit tedious. So we were stacking our segmentation masks into a volume, then applying marching cubes to extract the surface mesh, cleaning everything up with some spherical processing and smoothing, and the final step was to render and export to PLY format. So we go from extremely messed up data floating all around the space to a disc-like shape that was finally rendered into a 3D model of what is actually a mouse heart cell. Now what you see here are the actual 3D reconstruction from salivary and tongue cell nuclei. This isn't just a visualization trick though. We're recovering genuine 3D structural information that simply does not exist in traditional 2D analysis. For researchers studying tissue organization and cellular architecture, this represents a significant step forward in what's computationally possible.
So, as a result, here you see how our pipeline transforms a raw histology patch into a crisp cell mask, and a simple cleanup step removes stray speckles to produce smooth masks. Feeding these clean masks into our 3D reconstruction yields dramatic visual improvements. As you can see, downsampled input on the left is rough and full of holes, whereas the SR enhanced input on the right is polished, artifact-free, and captures fine surface details. Quantitatively, as a result, noticeable improvements can be seen, and for example, super-resolution pre-processing cuts reconstruction error or Hausdorff distance by 25% and raises overall mesh quality by 20%. Key takeaways of the projects are: first, SR pre-processing sharply improves segmentation mask quality. Second, cleaner mask yields smoother, artifact-free 3D reconstructions. Third, the SR to Unitree workflow is modular and easily adaptable to other tasks. And the last is open source code, and the models ensure full reproducibility to anyone who wants. Thanks for your attention.
Jumping back in really quickly just to explain the work division. So me, Rita, I worked on the data pre-processing and the 3D reconstruction. Zainab, she worked on the super-resolution models testing and application. And Jaris Bek handled the segmentation models and the application results.